transport, which is very important in physiology and also in our body physiology mechanisms. And to understand the medicine, to be a good practitioner, we need to learn about the membrane physiology, membrane transports. As we have learned in the previous class about the structures of cell membranes, structure of the cell membranes, today we can see the picture at first. Okay, before going to the next class, I will ask some questions from the previous class. Did you gone through with the class I have taken? Did you gone through?
Yes, sir. Noted.
Yes, sir.
Okay, I will ask some questions from previous class. Am I clearly audible?
Yes,
Yes,
sir.
sir.
Okay. Going to read the cell connections. Please show the slide. Uh, First slide, cell connection. Can you see the first slide? Can you see the first slide?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So this is the connections of cells, mainly between two cells. Um, Cells have all those connection to export and import the substances to each other and maintains the homeostasis mainly. Um, the intercellular connection, that there's two types of junctions between the cells that makes up the tissues. Junctions that fastens the cells to one another and to surrounding tissues, like tight junctions, that is also called zonula occludens. You should remember the tight junctions. You can see the tight junctions here, which are which are attaches tightly or closely, more closely, is the tight junctions. These types of junctions tie the cells together and into tissues with strength and stability mainly. And we have desmosomes and zonula adherens, which hold cells together. You can see the desmosomes and hemidesmosomes also. Hemidesmosomes, those you can see, and focal adhesions attaches cells to their basal laminus mainly. Basal laminus means the structure with which the cells are remains attached mainly. So basal lamina, that means at the base where the cells are sits or cells positions, they attaches with the lamina or base, with their base, by hemidesmosomes. And there is also a gap junction. So remember that about the tight junction and gap junction mainly. Tight junction and gap junction. Tight junction have all those desmosomes, semidesmosomes, and mainly the tight junction proper, that means zonula occludens. And gap junctions is different one. The junction by which molecules are transferred is the gap junction mainly. Molecules are transferred by gap junction. Am I clear here?
Yes, sir.
Okay. Now, please show the next slide, please. Oh, these are also same slide. You can see the cell junctions. Please show the previous one. I'm explaining this one. Cell-cell junctions, tight junction, adherent junction, desmosomes, gap junctions. You can see, and we have cell matrix junction, in which the extracellular matrix and actin-linked cells matrix junctions also. Please show the next slide. Junctions and components, you can see tight junction has components like as I said, jonula occludins. So there is occludins, there is clodine proteins, junctional adherent molecules. There is adherent junctions, co-adherent adhesion receptors, cytoplasmic proteins. Cap junction has connecting transport membrane proteins, and desmosomes have co-adherent family proteins like desmo desmoglin and desmocolin. You don't have to remember all those things. Mainly just remember the tight junction and gap junctions. Okay, that's all for you. And please show the next slide, please. Okay, now we're going to the membrane transport. But before that, I'm explaining the intercellular connections. Intercellular connections about the gap junctions mostly. Gap junction is important. It is type of intercellular connection adjacent cells connect with each other by via channels, via tunnels, as you can see in the pictures. Please show the previous slide, please. Azim, show the previous slide, please. Previous one. The previous one again. Previous to this. Okay. So you can see the structure of gap junctions here. The gap junctions, the intercellular space narrows from 25 nanometer to 3 nanometer, and the units called connect zones. Connect zones in the membrane of each cell are lined up with one another. Each connection is made up of 6 protein subunits called connexins that we are saying that the content of gap junction. They surrounds a channel that when lined up with the channel in the corresponding connect zone in the adjacent cell permits substances to pass between the cells without entering the extracellular fluid. The diameter of the channel is normally about 2 centimeter, which permits the passage of ions, sugars, amino acids. Remember, what are the what they permits? The gap junction permits the passage of ions, sugars, amino acids, and other solutes with molecular weights up to about thousands. So it is the type of intercellular connection. Adjacent cells connect with each other via tunnels, and through these tunnels, materials from one cell can pass into other without having to go into the extracellular fluid. And functions is, as I said, permits the passage of ions, sugars, amino acids, and other solutes with molecular weights up to about thousands. And gap junction permits the rapid propagation of electrical activity. Remember, rapid propagation of electrical activity from cell to cell. And number 3, it exchanges the various chemical messengers. Exchange of various chemical messengers also done by this. So number 1 permits the passage of ions, sugars, amino acids, and other solutes. Number 2, gap junctions permits the rapid propagation of electrical activity. And number 3, exchange of various chemical messengers. That's all for the functions of gap junctions. And intercellular communications. In intercellular communication, it is a sophisticated communication system to coordinate the functions of the cells, tissues, and organs by a variety of regulatory signals to maintain the internal environment for maintenance of life. As we say, that homeostasis. Everybody tries to maintain the homeostasis, that is the nearly constant condition. comparison between the intercellular communications, if we compare with the gap junctions and synaptic junction, the do you know what is the synaptic junction? Synapsis.
The junction between two neurons.
Exactly, junction between the two neurons or neuron to muscles, or yes, connection between the two neurons or neuron to muscles. That means it may be connected by the axon to dendron, or axon
Axon to
or
axon.
dendron to dendron, or axon to axon. All those are synaptic connections. So we have cell connections of gap junctions and neuron connection of synaptic junctions. We have paracrine and autocrine junctions also. These are other cell junctions. We can differentiate them. Message transmission that gap junction does is directly from cell to cell, and synaptic junctions does it by across synaptic cleft mainly. And paracrine and autocrine cells does it by diffusion and interstitial in the interstitial fluid. And gap junction is mainly does the transport locally. Synaptic also does by transport local transport, and in paracrine and autocrine cells also does they also locally diffuses. And specificity gap junctions specificity depends on anatomic locations, mainly gap junctions specificity. And synaptic specificity depends on mainly anatomical locations also, and receptor location also. That means where the receptors they connect, they get the signals specifically. And paracrine autocrine junctions depends on the receptors mainly. Specificity depends on receptors. Am I clear here?
Yes, sir.
Okay. Uh, okay. Before going to the next slide, uh, I said Berliner to explain about the functions of membrane. Now we'll go to the transport system, mainly after the cellular communication. Go to the next slide, please. Yes. So transport system. Transport of substances through the cell membrane is the membrane transport. As you know, what is membrane transport? If you asked what is membrane transport, membrane transport is the process by which different substances are transported across the biological membrane in a biological system. As simple it is. So, types of membrane transport, if you asked the types of membrane transport, you will say membrane transport is two types. Sir or madam, this membrane transport is two type. One is passive transport, another is active transport. Passive transport has three brothers. One is simple. Now we're going to know about the cell transport. As I was saying, that we have two transport system. One is passive transport, and another is active transport system. One is simple diffusion, another is osmosis and facilitated diffusion here. So simple diffusion has so passive diffusion has three brothers: simple diffusion, osmosis, and facilitated diffusion. Active transport, passive transport has three brothers: simple diffusion, facilitated diffusion, and another is osmosis. As you have gone through in the higher secondary level about the osmosis and simple diffusion, facilitated diffusion, isn't it?
Yes, sir.
Yes, sir.
Okay. So it will be easy, but now in physiology it will be in details. And active transport have pump and vesicles. Active transport have mainly primary active transport system and secondary active transport system. And vesicles have exocytosis and endocytosis. Endocytosis has a pinocytosis, phagocytosis, and receptor mediated um, transport. Those are the classification mainly you have to remember. And the transport process, if you go with the exocytosis and endocytosis, there's the vesicle system. There's also inside the active transport. If you say about the exocytosis, there are mainly neurotransmitter, hormone, enzymes, proteins are released from the cells by this process, exocytosis. That means exo means out of the cells. So when it goes out, the neurotransmitter, hormone, enzymes, proteins are released from the cells. That means the exocytosis. That is done by the vesicles. Vesicles means capsules. Vesicles means capsules or pack that releases mainly neurotransmitter, hormones, enzymes, proteins, and other things. And endocytosis means the pinocytosis or phagocytosis. Pinocytosis, pino means pino means drinking. Pino means what?
Breaching.
Drinking.
Yes, sir.
That means pina in Hindi. Always occurs down a concentration gradient. These are the passive transport. What do you mean by concentration gradient? What do you know about the concentration gradient? Do you know what is concentration gradient?
High potential to low potential.
Yes, this is a concentration gradient. Um, Not only a high concentration to low concentration.
Hypotonic isotonic.
Difference between the concentration. These are the concentration gradient. Always maintaining the concentration gradient. Okay. Sometimes diffusion. That is the opposite of osmosis. Osmosis occurs from high concentration. From low concentration to higher concentration, and diffusion occurs higher concentration to low concentration. So both may occur in the passive transports. So concentration gradient is the two mediums maintaining the concentration, and we have facilitated diffusion also. We'll discuss in details in the next. And please show the next slide. So movement of the molecules across the membrane along with the concentration gradient, that is the passive terms. So you can see the concentration gradients now. A difference in the concentration between two areas is called a concentration gradient. What effect might the gradient have on speed or diffusion? That is the question. When you have a higher concentration in one gradient and low concentration in one gradient, so the difference is the concentration gradient. Please show the next slide, please. We'll discuss about the speed in the next lesson. Okay, diffusion. So we have passive transport in three forms. One is diffusion, another is osmosis, another is facilitated diffusion. So diffusion, the net movement of the particles from an area of higher concentration to the area of low concentration, as I said. Osmosis is lower concentration to higher concentration, and diffusion is higher concentration to the lower concentration. Can you give an example of diffusion? Anyone?
River water.
River water? Not really. The common example is a
Cold
perfume.
water and hot water.
Is a perfume or scent bottle. Perfume bottle. What it does? What it does? In a higher concentration in the bottle, and when it is sprayed or when it is in an open, when you open the tap, you will get the scent throughout the environment. That is the diffusion is doing itself. There is no force or nothing else for the diffusion. So if you if you do any force to make it as a diffusion, it will be facilitated. But in case of normal diffusion, it is doing it itself. So the net movement of the particles from an area of higher concentration to an area of lower concentration, this is the diffusion. Due to the random movement of the particles, this occurs due to the random movement of the particles. A passive process, which means that no energy is needed. That means if you use energy, that is facilitated. That is facilitated by that energy, but you are not using any energy. Okay, so it is doing it itself. So passive process. Am I clear here about the diffusion?
Yes, sir.
Yes, sir.
Okay. If you have any doubt, ask it instantly. Okay. Next one, please. Next slide, please. Okay. You can see the diffusion on of the screenshot here. Movement of molecules from an area of a higher concentration to an area of lower concentration. Same thing is saying in this lecture. Exchange of oxygens and carbon dioxide between the lungs and tissues. Yes, example in the body she is giving. Exchange of oxygen and carbon dioxide between the lungs and tissues. This is also diffusion. How can you explain? Anyone? Exchange of oxygen and carbon dioxide between the lungs and tissues. How it is diffusion?
Sir, lungs have oxygen-rich blood, and the tissues has carbon dioxide concentration more. So, carbon dioxide comes to the lungs, and oxygen goes to the tissues.
Just remember the mechanism of lungs. When you have carbon dioxide rich blood in your vessels in your body, it gets the signals to your respiratory centers, and your respiratory center is saying that carbon dioxide level is increasing, so please inform the diaphragm. As you know, in the anatomy, thoracic outlet has diaphragm and intercostal muscle that maintains, that controls the lungs expansion and contraction. So it gets a signal to it gives a signal to the thoracic muscles and diaphragm, so that they contracts and expands the thoracic cavity, so that lungs may expand. And when lungs expand, the the concentration of gases inside the lungs is decreasing or increasing.
Increasing.
How? You are expanding the lungs, so you are not taking the gases instantly. First of all, you are expanding the lungs. Only that time, which what occurs? Only that time you have all normal level of gases in the lungs, but the lungs is expanding, so its space is increasing. Space is increasing, so that the gas is now reducing its concentration. You are not increasing or increasing the gases. You are expanding the space. Am I clear? You are expanding the space. That means lungs, but gas is the same level. Your concentration of gas is same. Sorry, your level of gas is same, volume of gas is same, so the concentration is reducing here. Pressure is reducing here in the lungs, but atmospheric pressure is higher now. In the lungs, the pressure is decreased because of your expanding your lungs. So the atmospheric pressure is increased now. So the concentration of your atmospheric is increased so that they can enter into the lungs by diffusion. So you can inhale the lungs. If lungs concentration was greater, then you couldn't take inhalation. You couldn't inhale gases because atmospheric pressure may lower. So it occurs in case of exhalation. It occurs in case of exhalation when your concentration of lungs lungs gases your concentration of gases in the lungs increases and atmospheric gases concentration decreases. It helps you to exhale the gases. Am I clear here?
Yes,
So
sir.
exchange of oxygen and carbon dioxide between the lungs and tissues is also done by diffusion. Am I clear, or do you have any doubt?
Sir,
No,
what
sir.
about lipid soluble substance transported across membrane?
Sorry, sorry,
Sir,
say it again.
for lipid soluble substance transported across cell membrane.
The question I asked previously, it didn't ask
Yes,
the
sir.
question then. I said when you have a question or any doubt, ask it instantly. Okay. Lipid soluble substances are transported across the cell membrane by diffusion through lipid bilayer diffusion also, and lipid soluble substances are transported into the cell because they are already lipid bilayer. They are allowing their transport lipid soluble substances as it is soluble. You are saying it lipid soluble. That means it is solute. It can solute itself in inside the lipid. Are you clear?
Yes, sir.
Okay. When you have you are getting a higher concentration to lower concentration outside, if you have outside the cell a higher concentration of lipid and inside the cell lower concentration, then you can easily diffuse into it. Okay, that is the relation between the diffusion and lipid soluble substance. And facilitated diffusion substances are transported by diffusion through a membrane using specific carrier protein to help. As I said, in case of only diffusion, you don't need, or a diffusion proper, you don't need any carrier protein. But it is facilitated diffusion. You need a carrier protein to take the transported substance inside the cell. We'll get it in the picture next slide inshallah. And diffusion of glucose gets some of the carrier protein. These are the facilitated transport. This is the example of facilitated diffusion. Some of the carrier protein like GLUT1, 2, 3, 4 are the example of facilitated the example of carrier protein who transport the glucose inside the cell. These are the example of facilitated diffusion. So we have all those example of diffusion. Am I clear here?
Yes, sir.
Okay. If you have any doubt, you can ask instantly. Okay. So passive transport. You can see the passive transport picture here. Simple diffusion through the lipid bilayer. This is the process how lipid soluble substances transported itself from outside to inside the cell, because when you have outside the higher concentration, you can see in the picture in the first one, outside to inside. When you have higher concentration in outside, you're getting easily diffused inside the cell through the plasma membrane. And you have non-specific transporter facilitated diffusion. You have carrier protein, that purple one. You can see the carrier protein through a non-specific transporter, and you have specific transporter, especially for glucose. As we have learned in the glucose transport as an example, glucose have a specific transporter, that is GLUT. We have GLUT 1, 2, 3, 4. These are the specific carrier protein that transport only glucose. Facilitated diffusion through a specific transporter. Am I clear here?
Yes, sir.
Okay.
Yes, sir.
So you both may have
Yes, sir.
you may have both non-specific transporter and a specific transporter in case of a specific carrier protein or non-specific carrier protein. And simple diffusion have a just simple diffusion through the lipid bilayer. And we have osmosis through the lipid bilayer and then anoprotein. In the right picture, you will get it next in how the osmosis occurs. Please show the next slide. Diffusion across the plasma membrane. Picture is showing the same thing. Fat soluble molecules you can see, and channel protein, water soluble molecules you can see, and how the fat soluble, how one asks how the lipid soluble can transfer. This one, this picture shows you the how lipid soluble. Lipid means fat, so fat soluble and water soluble. Yes, factors affecting the rate of diffusion. Some factors affects the rate of diffusion. You may ask in the viva, and you may ask in the written also. Size of the particles. When size of the particles is bigger, it takes the more time in case of diffusion. At a given temperature, a smaller particle moves or diffuse faster than a larger one. So smaller, it gets faster. And temperature. As the temperature increases, particles gain energy and move faster. It moves randomly. Thus, the rate of diffusion is increased. So, increased temperature increase the rate of diffusion. And concentration difference. When you the greater the concentration difference between the two regions. So, when you have a bigger concentration gradient, you'll get more diffusion or faster diffusion. The faster substance will diffuse. Then diffusion distance. At a given temperature, it takes longer for particles to diffuse farther distance. That's the slower the rate of diffusion. The surface area. The greater the surface area, the greater the rate of diffusion. If you have low surface, small surface area, you have small diffusion rate. Permeability. The more permeable the separating surface is, the faster a substance can diffuse through it. Are you clear here?
Yes,
Yes,
sir.
sir.
Okay. Go to the next slide, please. Okay, this is the curve rate of diffusion shows by this chart or this graph. Concentration of the substance is in the axis area, and the rate of diffusion. And you can see simple diffusion is expressed by blue one, and facilitated diffusion is expressed by the red one. This is the Vmax. Vmax, that means velocity maximum chart or velocity maximum graph. You can see the simple diffusion. It is going upwards very quickly. Rate of diffusion is simple diffusion, and in case of facilitated diffusion, it goes upwards at a maximum rate, then it goes flat. So that maximum velocity of the facilitated diffusion is called Vmax, the velocity maximum. Are you clear? Are you clear here?
Yes, sir.
Okay. The diffusion of solvent molecules across the semipermeable membrane from the region of lower solute concentration to the region of higher solute concentration is called osmosis. So we are on osmosis now. The diffusion of solvent molecules across the semipermeable membrane, as I said previously, that lower concentration to the higher concentration. Difference is lower concentration to the higher concentration. So osmosis, what is the example in our body? Osmosis of water from the renal tubules. Osmosis of water, as we know, reabsorption of water occurs in our renal tubules and nephrons. When we'll learn about the renal system, we'll get it, inshallah. The osmosis of water from the renal tubules is also another one. Osmosis of water from the renal tubules, the example, reabsorption occurs in the renal tubules. So osmotic pressure. What is the osmotic pressure? You may ask in the viva, what is the osmotic pressure? The exact amount of pressure required to stop the osmosis is called the osmotic pressure. When you need to stop the osmosis, the pressure plays the role of the pressure plays the role. So it is osmotic pressure. The osmosis and diffusion. You can see the picture of difference between the osmosis and diffusion. You will get the chart also in the next slide, but now you can see how osmosis occurring from the higher concentration to lower concentration and lower concentration to higher concentration. This is osmosis. As I said, it passes of solvent from a dilute solution through a semi-permeable membrane. You must need a semi-permeable membrane. You must need a semi-permeable membrane. That's why I added this slide, a semi-permeable membrane into a more concentration, because if you want to make a osmosis, you must need a semi-permeable membrane. What is semi-permeable membrane? A thin layer of material that allows certain types of molecules to pass through while prohibiting other molecules from passing through, dependent on the size and geometry of the molecules. Some semi-permeable membranes allows the high increased size molecules, and some allows lower size molecules. So depends on the size and geometry of the molecules also. Shape of the molecules also depends. Shape of the it also depends on the shape of the molecules. So osmosis, to investigate the osmotic effects of different concentrations of the sugar and salt solution, to use all practical skills we have developed, data recording, presenting and analyzing and evaluation of the results. So this is a practical use of osmosis, mainly to use data to deduce the osmotic content of the cell. So to investigate the effects of different concentration of sugar and salt solution in our body is maintained by the osmosis. So is maintained by learning about the osmosis. Okay, next one please. So difference between the osmosis and diffusion here, very important. It is limited only to the liquid medium and occurs limited only to the liquid medium. Osmosis occurs only the limit only the liquid medium, and diffusion occurs in all mediums, solid, liquid and gas, all mediums. Requires a semi-permeable membrane. Obviously you need a semi-permeable membrane in osmosis, but you do not require a semi-permeable membrane in case of diffusion. Both simple and facilitated, you don't need any semi-permeable membrane. And osmosis depends on the number of the solute particles dissolved in the solvent, and diffusion, if in diffusion you need to depend on the presence of other particles, and osmosis requires water for the movement of the particles. Diffusion, you do not need require the water for the movement of the particles. Only the solvent molecules can diffuse in case of osmosis, and in case of diffusion both the molecules of solute and solvent can diffuse, because as we said previously, the solid, liquid and gas all can pass through the diffusion. Just you need higher concentration to lower concentration. So concentration gradient. The flow of particles occurs only in one direction in case of osmosis, but flow of particles occurs in all direction. Just you need a concentration gradient of higher concentration to lower concentration. That's all for the osmosis versus diffusion. Please go to the next slide, please. This is also difference between the osmosis and diffusion. You can see some other differences. Flow of either solute or solvent molecules in the both direction, as we said, as we saw in the previous slide. It involves flow of solvent molecules in one direction. Both direction for diffusion, osmosis for one direction, and no such membrane is required. It is requires the semi-permeable membrane. It is the faster process, fast process. This is the new one. It is a fast process in diffusion, and slow process is osmosis. So which one is the slow? Response, please. Which one is the slow?
Osmosis.
Osmosis. The faster is diffusion. Okay. Occurs in the liquids as well as gases, and this is occurs in the liquids only. Osmosis. The movements are over the large distance, and the movements over the short distance. Osmosis. Next one, please. Active versus passive transport. Show the picture, previous picture, please. Show the previous picture. Active versus passive. You can see the active transport versus passive transport. You can see the difference of active transport, passive transport. As diffusion occurs higher concentration, lower concentration, no need of energy here. But active transport, you need energy to initiate the transport system. ATP is working here. You can see the ATP plays a major role here. That's why it is called active transport. So you will be asked that which transport need ATP, or which transport need energy, and which transport don't need energy. You will say passive transport don't need energy and active transport needs energy. Please show the next slide. This one, the active transport. Active transport is divided into two types. So we have gone through passive transport all, and now it is active transports time. So active transport is divided into two types according to the source of energy and used to cause the transport. This is primary active transport and secondary active transport. Please show the next slide, please. Primary active transport and secondary active transport. The active transport, as we said, requires use of energy. The characteristics of active transport requires use of energy because it doesn't happen naturally through the diffusion or facilitated diffusion. A kind of facilitated diffusion. Facilitated diffusion, we need carrier protein, but in this case we need energy also. So requires use of energy because it doesn't happen naturally. If you don't have energy, you cannot. There will be no active transport. So naturally through the diffusion or facilitated diffusion, it occurs. Requires a carrier protein that uses energy from the ATP molecule. So you both need carrier protein and ATP. As we saw in the facilitated diffusion, we need carrier protein, but in case of active transport, we need both carrier protein and ATP molecules, that is energy, adenosine triphosphate, a nucleotide that stores energy and can release it when bonds are broken. So when the ATP is broken, there's a huge energy releases. That energy helps you to transport the substance from one concentration gradient to another. So why moving stuff against the concentration gradient requires a pump? Why it needs a pump? We'll see next, inshallah. Or fasting or rebuilding of cell membranes, fusing or rebuilding cell membranes. We'll learn it next. Okay, next slide, please. Active transport, as I said, process of moves materials across the plasma membrane requires energy from the cell. From the ATP, and materials move against the concentration gradient, low concentration to higher concentration. Three kinds of active transports. One is pumps, one is endocytosis, one is exocytosis. So active transports are those, as we saw in the classification. Please show the next. Primary active transport, as I said, that we have two active transport system. One is primary active transport and another is secondary active transport. So primary active transport is this, requires the direct expenditure of energy in the form of ATP in order to move ions against an electrochemical gradient, and in turn can generate a voltage across the membrane. So example is sodium potassium ATPase pump is a very important pump. It occurs all over the body to maintain homeostasis. Potassium ATPase pump is very important. Don't forget about the sodium potassium ATPase pump, because it occurs throughout the body and occurs in all cells almost. So we have hydrogen ion pump also, is hydrogen ion ATPase pump also, that generates voltage as it transports positive ions in one direction. Positive ions, that means proton. So this is also primary active transport. Let's show the next slide. Primary and secondary active transport, as I said, primary and secondary active transport. In primary active transport, the energy is derived directly from the breakdown of ATP. In the secondary active transport, you will this slide I added because you when I said the primary active transport, you must have a question about the secondary active transport. Then what secondary active transport uses? So in secondary active transport, the energy is derived secondarily from energy that has been stored in the form of ionic concentration differences between the two sides of the membrane. So energy stored previously, that is used in later, that is called secondary active transport system. So please show the next one. So secondary active transport in details. Secondary active transport, also called co-transport, also uses ATP, and but one substance uses ATP to cross the membrane while another sneaks it without directly using ATP, like a revolving door. And there are two main forms of secondary active transport: antiport and symport. Antiport is the two different molecules are pumped in opposite direction. Two different molecules pumped in opposite direction. That is antiport. That's mean exchange pump. And symport is co-transport. That means uses the downhill movement of one solute species from high to low concentration to move another molecules uphill. One goes downhill and one goes uphill from low concentration to higher concentration. That is symport. The same molecules transports in the symport, and antiport is the different molecules transported. So this is the secondary transport, so where the ATP is uses stored ATP is used. So as we are in the secondary active transport system, please show the next slide. Next slide. Okay, this one, the energy is derived secondarily from energy that release in simple diffusion. That means when you are asked that where the secondary active transport get energy, you will say that stored energy, ma'am or sir. Then she or he will ask that from which stored energy. You will say secondary active transport and the co-transport. The energy is derived secondarily from energy that release in simple diffusion. And secondary active transport, co-transport, and secondary active transport in case of counter transport, this also gets energy as stored energy. So those are used in secondary active transport mainly. Okay, please show the next slide. So this is the primary active transport and secondary active transport picture. You can see the ATP is driven sodium potassium pump stores energy by creating a steep concentration gradient of sodium entry into the cells. You can see sodium potassium pump. See the picture closely. You can see the extracellular fluid and intercellular fluid. Inside the cell, it is cytoplasm. Intercellular fluid outside. You can see the sodium potassium pump. When ATP is creating an energy, ATP is broken. ATP is broken, and ATP creating adenine diphosphate, that means ADP, and an inorganic phosphate, and creates a lot of energy, huge energy, by dissociate itself, and this energy pushes the sodium and potassium pushes the sodium and potassium to binds into their binding site, bind with their binding site, and they um, puts outwards to outwards three sodium ion, and it gets inside the two potassium ion. Three sodium ion goes out, and two potassium ion goes in. So there is always a one electron deficiency. Always a one electron deficiency. Two potassium gets in, and three sodium gets out. Three sodium gets out, two potassium gets in. So there is always a deficiency of one electron. So there is always a negative charge is maintained outside the membrane, and inside the membrane always a positive charge maintained. The secondary active transport we have as a sodium diffuse back across the membrane through a membrane co-transporter protein, you can see, and it derives glucose against a specific, and you can see specific against the concentration gradient into the cell. The sodium glucose symport transporter loads the glucose from the extracellular fluid, and sodium glucose symport transporter release glucose into the cytoplasm. You can see. Are you clear here?
Yes, sir.
Here is the membrane, plasma membrane, the sodium potassium ATPase pump. You can see, I explained, ADP is divided, ATP is divided into ATP, and there is a huge level of energy releasing, and they helps the three sodium to go out, and they helps the three sodium to bind with their binding site, and pushes them to go out, and they also creates a concentration gradient so that 2 potassium goes inside the cell. So please show the next slide. Secondary active transport, as you can see, low sodium glucose, high sodium outside, and the higher concentration to lower concentration, they're coming. Energy releasing transport system. So glucose is that shape, you can see, diamond shape. Okay. The criteria of active transport, you must need against you must have to work against a gradient. You have to work against a gradient to make active transport, and membrane must be present. You must have a membrane here. Your energy is necessary, your carrier protein is necessary. You need all those things. So you need carrier protein, you need energy, you need a membrane, and you must have to work against a gradient. So this is the active transport. Please show the next one. Importance of active transport to control the cell volume, as if you don't maintain the negative pressure outside and inside the positive uh, always, then water will enter into the cell and it will get swell, it will blast the cell. So maintain the cell volume and generation of the membrane potential. It maintains the membrane potential always. The sodium potassium ATPase pump maintaining the membrane potential and controlling the cell volume. Am I clear here?
Yes, sir.
Do you know how membrane potential is maintained?
No, sir.
The electrical potential difference across the membrane is called membrane potential mainly. Electrical potential difference, as I said in the ATPS pump, you have two potential system, resting membrane potential and action potential. We'll discuss in the next class, inshallah. So about the membrane potential and action potential, how potential works for the cell, and that's all for the secondary active transport. And basic mechanism of transport of sodium ion across an epithelial sheet with the um, diagram you have seen already. Basic mechanism transport of sodium ion across a epithelial sheet is at many places in the body substance must be transported all the way through a cellular sheet instead of simply through the cell membrane. Transport of this type of occurs through the intestinal epithelium, epithelium of the renal tubules, epithelium of the exocrine glands, and epithelium of the epithelial cells of the gallbladder, and of the membranes of the choroid plexus in the brain, and other membranes. The basic mechanism for transport of substance through a cellular sheet is active transport, as you know, through the cell membrane on the one side of the transporting cells in the sheet, and then either simple diffusion or facilitated diffusion through the membrane on the opposite side of the cell. So process of process of sodium transport is this. And we have a question about electrogenic nature. The electrogenic nature of sodium potassium pump. You may ask that describe the electrogenic nature of sodium potassium pump. As I explained previously, the fact that the sodium potassium pump moves three sodium ions to the exterior, and for every two potassium ions mainly, if you give two potassium ion inside interior, you will get three sodium ion. That a net of one positive charge is net of one positive charge is moved from the interior to the cell to the exterior. So for each cycle of pumps, that this creates a positivity outside the cell. I previously said that creates a negativity outside the cell. That was wrong. As I'm saying that it creates a positivity outside the cell because there is three sodium, that is positive ion, and two potassium inside. So it creates a positivity outside the cell. So this creates a positivity outside the cell but leaves a deficit of positive ions inside the cell. The one deficits because it gets only two potassium and gets out of and three positive ion is gone. So that is, it causes negativity on inside. Negativity on the inside, not outside. Negativity inside. Therefore, the sodium potassium pump is said to be the electrogenic because it creates an electrical potential across the cell membrane. Importance is electrogenic pumps creates electrical potential that is basic requirement in the nerve and muscle fibers for generation and transmission of nerve impulse and muscle contraction. So how important this is? If you don't get any signals or you don't get any impulse by the nerve fibers, or if your sodium potassium pump is stopped, you will not get any generation of impulse or any transmission of nerve impulse. So your respiratory system, your all the systems will be blocked, paralyzed if you don't have any sodium potassium pump. The sodium potassium pump so maintains our homeostasis. Importance of sodium potassium pump for controlling of cell volume, you'll be asked in the viva. Inside the cell, do you have any question or any doubt here?
No, sir.
You may be asked, the sodium potassium pump controls the cell volume. How the sodium potassium pump may control the cell volume? Do you know?
No, sir.
Suppose one of the most important functions, the one of the most important function is sodium potassium pump is to control the volume of each cell. Without function of this pump, most cells of the body would swell until they burst. As I said previously, the mechanism for controlling of the volume is inside the cells. Suppose inside the cell are large numbers of proteins, isn't it?
Yes, sir.
Inside the cell, there are a large number of proteins and other organic molecules also that cannot escape from the cell, isn't it? They cannot escape from the cell. So most of these are negatively charged, and therefore, most of the cells are negatively charged inside the cell as sodium potassium pump is activating. So most of the cells are negatively charged and therefore attract large number of potassium from outside. It attracts large number of potassium that's coming from outside, and also it attracts sodium and other positive ion also as well. So all these molecules and ions, this cause osmosis of water to the interior of the cell. Unless this is checked, because always there is negative potential inside the cell, and may attract all those positive ions. Unless this is checked, the cell will swell indefinitely. It will swell and swell again and again indefinitely until the cell bursts. So the normal mechanism for preventing this is the sodium potassium pump. It gives expel out the sodium ion as positive ion to outside, and gets only two potassium ion inside, so that it always at the cell surface always there is a positivity maintaining. If there is negativity outside the surface, it will take all those water inside and it will swell and it will burst. Am I clear here?
Yes, sir.
Okay.
Yes, sir.
The membrane is far less permeable, so sodium ion can then sodium ions, the membrane is far less permeable to sodium ions, as you know, than to potassium ion. So that once the sodium ions are on outside, they have a strong tendency to stay there. Thus this this represents a net loss of ions out of the cell, and which initiates osmosis of water out of the cell as well. So it is doing both. It is preventing to coming of water, or in entry of water, and it is also expelling some of the waters outside. And if a cell begins to swell for any reason, this automatically activates the sodium potassium pump. Suppose you have any foreign body or a disease condition and you are getting your cell swelled, so this automatically activates the sodium potassium pump, moving still more ions to the exterior and carrying water with them. Am I clear?
Yes, sir.
So this is the importance of sodium potassium pump for controlling the cell volume mainly. Can you reply when you are asked? When you will asked? Will be asked.
Yes, sir.
Okay. So importance of sodium pumps. The sodium potassium pump inhibitor is used in a heart failure, and it is also used as spironolactone. Spironolactone is a diuretic that creates more of the urine, and it inhibits the sodium potassium pump and causes natriuresis. That means sodium gets outside. It creates a natriuresis, I will. It's not needed natriuresis, but the clinical. When you are saying the when you are asked that what is the clinical importance of sodium potassium pump, you will say sodium potassium pump inhibitor is used in a heart failure mainly. Is the clinical importance, and physiological importance is responsible for the generation of membrane potential and control of the cell volume, as I explained. Basis of nerve and muscle function, as I said. If you don't have sodium potassium pump, you will not get any signals. Provides energy for the secondary active transports mainly, as I said. So that's all for the active transports and membrane transport system. Thank you all for attending the class.